Welcome, everyone. I'm Lynn Chang. I'm Vice Chief of the Division of Digestive Diseases at UCLA, and I have the privilege of introducing our CTSI speaker, Dr. John Carethers. He's Vice Chancellor for Health Sciences and Distinguished Professor of Medicine at University of California, San Diego. He previously served as Chief of GI at UCSD for 5 years before he moved to Ann Arbor. And I learned today that he was born in Detroit. And he moved to Ann Arbor to become the Chair of Medicine at University of Michigan for almost 14 years before returning to San Diego. Dr. Carethers is a highly regarded gastroenterologist, renowned for his research on colorectal cancer, particularly focusing on the underlying genetic and molecular mechanisms. His work has significantly contributed to understanding the disparities in colorectal cancer incidence and outcomes among different racial and ethnic groups. He has identified key genetic mutations and pathways involved in the development and progression of colorectal cancer, helping to pave the way for targeted therapies and personalized medicine in this field. He has published extensively in peer-reviewed journals of high impact and served in leadership roles in various professional societies. This includes that he was president of AGA, the American Gastroenterological Association, which is our premier GI society organization in the country. And all his work has contributed to the advancement of gastroenterology and oncology. He's had multiple federal grants from the NIH, VA, and they also include training grants, which I know that he's very passionate about. He's authored over 200 papers, as I said, published in high impact journals such as New England Journal of Medicine, Gastroenterology, JCI, among others. And from a personal standpoint, as he mentioned to me today, he's a people person, which you will obviously see, and he's probably one of the most charismatic leaders that anyone could ever meet. And he's intelligent with a high EQ, and he really exudes positivity. So we're really very grateful to have him speak to us today.
Thank you, Dr. Chang. You're too kind to me. It's a pleasure being here, and I want to remind everyone that March is Colon Cancer Awareness Month, so it's apropos that I'm here in March. And in the setting of soon, in 2 weeks, the American Association of Cancer Research, which Dr. Rebus in the audience, who was president of AACR, their conference starts in 2 weeks down in San Diego. So just down the road, if you ever want to just say hi to me too, and I'll hopefully host you as well. So it's my pleasure being here. It's the first time I'm actually giving a talk at UCLA. And I really want to thank Dr. Rebus and the CTSI for inviting me here. So my title of my talk is Inflammation and DNA Mismatch Repair in Colorectal Cancer. And these are my disclosures. None of them have related to anything I'm going to talk about today at all. So my outline is going to be just talking just in general what's high level for inflammation colon cancer, and then go into DNA mismatch repair. Many of you might know a lot about microsatellite instability, so I'm going to just do a high level on that, and then talk about EMAST. Many of you may not know what EMAST is, and I'll tell you some of the things that's going on there, and we're still discovering things. And then a linkage to the microbiome and trying to understand how inflammation works there. As with most cancers, it's a gene-environment interaction, and studies before have said the environment, it may be even the bigger driver, even though we can have Mendelian inherited diseases, as well as multiple genetic contribution to cancer. So any genetically susceptible individual, 8 billion people in the world, we have different SNPs and different ways of interacting with our environment, and that can modify your risk for cancer. And certainly in the field of colorectal cancer, I think of this as, tumor initiation, tumor progression, because generally what happens in the colon is you go from normal mucosa to the earliest forms of an adenoma, which is a precancerous polyp that can progress, and with the right genetic changes, turn into something malignant. And that is highlighted by the genetic diseases, for instance, when something called familial adenomatous polyposis, FAP, where tumor initiation, those polyps are greatly accelerated, or something like Lynch syndrome, where the polyps still happen at a normal rate, but the tumor progression is accelerated based on the genetic defect. But in any of these stages, at either at tumor initiation, tumor progression, and even into metastases, there's different levels that can modify those rates that are probably contributed certainly by inflammation and certainly the microbiome. Now, this just highlights some of the risk factors for colorectal cancer. I think of these as non-modifiable risks versus modifiable risks. The non-modifiable things are the things you cannot change. You know, if you're born with something, your genetic background, or your age. You can't change your age. And if you can, let me know. We'll work on that. But then there's the modifiable things that in theory could alter that initiation or progression. And these are things like that's epidemiologically linked: diet, microbiome, tobacco usage, etc., hormone replacement therapy. The only thing that so those things are all controllable. They're very hard. They're very hard to do because they are lifestyle changes that must last for a long time. The thing that most physicians or practitioners can control is the screening utilization rates. And there's evidence that certainly screening for colon cancer can certainly modify your risk by removing these polyps. But these two things combined are the ultimate risk. And except for the screening risk, the utilization rates, I would argue that the environmental susceptibility all are related to inflammation in the colon. All those things, use of hormone replacement therapy, physical therapy, your diet, all relate to the level of inflammation in your colon. This is the landmark study published in Nature that highlighted the genetic changes seen in colorectal cancer from the DGSOM data set. So this summarizes about 30 years of genetic changes. These are somatic genetic changes that happen in the tumor. And in the upper left, just to walk through this very quickly, let's see if this laser works or not. It does. In the upper left is the y-axis shows the mutation rate, and the x-axis shows some characteristics of the tumors. To highlight here, you'll see the mutation rate is very high in this 15% of tumors, and then drops to sub 10 in the majority of colorectal tumors. These 15%, approximately 15%, are what we call hypermutated because they have a lot of mutations per megabase of DNA. And if you see here, microsatellite instability status, CpG island methylator phenotype status, which methylates one of the mismatch repair genes, so overlaps with MSI status, and silencing of MLH1 by methylation are all in this hypermutated group, whereas most of that is not in the non-hypermutated group. So these will have tens to hundreds to even 1,000 mutations in their genome, whereas the most common form of colon cancer has just a handful that drive it. And that is also seen in the downstream somatic mutations that as a result of the pathway of that tumor occurs. So for hypermutated tumors, you'll see most of these genes actually have coding microsatellites. So they shift in frame and take out anywhere from 25 to 75% of these very genes that most of these have coding microsatellites. This is unlike the 85% with the few mutations, where only a handful of mutations, the APC, P53, KRAS, TTN, and PI3 kinase, drive the majority of those colon cancers. And then you have a long tail of unique mutations that can self-identify as a fingerprint individual tumors from an individual person. So the hypermutated tumors, the pathway is very driven by loss of mismatch repair, but very common mutations. Most of these have a coding microsatellite, but in the non-hypermutated, only 3 or 4 genes drive the tumor with a long tail that are individualized to the tumor. And that's how colon cancer is driven genetically. This is easily put together in a little pathway. This is what Barbara Young and I wrote a few years ago on what I'll call the Burt-Volgenstein model or chromosome instability pathway, which is highlighted by the classic FAP driver, where you have a normal colonic stem cell, you lose APC or Wnt signaling, and you drive into this adenoma and colorectal cancer and ultimately into metastatic colon cancer, but it accumulates aneuploided because there's chromosome breakage and loss. That's why it's called chromosome instability. The other 15% is the microsatellite instability, and this is, you still have to lose Wnt signaling, but you typically, for most sporadic tumors, you methylate the MLH1 gene, one of these mismatch repair genes, and about 25% of them will have BRAF mutation, but it's really the accumulation of these frame shift mutations, like in the previous slide I showed you, that really drives it toward cancer and metastatic cancer. There's the CPG-island methylator phenotype pathway, which is, I'll say, is more prone to serrated adenomas, which are high-risk cancers, high-risk adenomas that can turn to serrated cancers, but they overlap with both of these pathways. So that's why most of these are under these, and that's why this doesn't add up to 100%. But in reality, you accumulate methylated genes, of which one of them is often MLH1, and then falls into an MSI high pathway. The outcome of these are different too. MSI pathway, which I'll show you in a few slides, if you have an MSI high pathway, that genetic defect drives inflammation, which protects the ability of the tumor from spreading. And that's why that's a favorable. This chromosome instability model doesn't have that same favorability, and their outcome is poor. And most of the CPG-island methylator phenotype, it's a more aggressive, even with MLH1 because of the other methylated genes, their outcome is generally poor as well. The other thing to mention on inflammation is the inflammatory milieu of the tumor can sometimes, and maybe even be a better predictor than classic staging of the tumor. This is a study from Jeremy Gallone, and he's published multiple papers on this, on immune typing colon cancers. And he'll look at the type of inflammatory cells that are at the center of the tumor, or CT, or at the invasive margin, IM. And with much better prediction than stage, he can show you if you have high CD3 tumors at the center of the tumor and invasive margin, irrespective of the classic staging, that he can predict a much better outcome than someone has low as compared to someone who's already stage 4 and has already had metastatic spread. So these lymphocytes can help predict with good accuracy the outcome of the patient based on the presence of that inflammation in the tumor. So let me go into DNA mismatch repair quickly. DNA mismatch repair, many of you know from either general biology or your genetics, that these are a series of evolutionarily conserved proteins that interact with each other to form heterodimers to repair usually single nucleotide mistakes in DNA, as well as frameshift mistakes at microsatellite sequences. You can have complete loss of mismatch repair by taking out MSH2, MLH1, or PMS2, or partial by MSH6 and MSH3, and I'll show you this in another slide. Most pathology departments now screen tumors by immunostaining and can help determine if they're going to be an MSI tumor, and or if they're a germline, and they can refer to your genetics group for germline testing. These proteins act as heterodimers. MSH2 pairs with 6 or 3, and MLH1 pairs with PMS2 in the human. They're reliant on each other. So if you lose one of the partners, the other partner becomes unstable in the cell. So this is why you can tell, like for instance, if you take out MLH1, its partner PMS2 is lost. But this is solely an MLH1 defect, such as in a sporadic MLH1 hypermethylated tumor. Obviously, when you take out mismatch repair, we don't measure the single base pair of mistakes, but we measure the microsatellite sequences, and that's what we call microsatellite instability. And that's what we can measure. And most microsatellites, there's about 100,000 of these throughout the genome, but only about 500 encoding regions, which is really driving the behavior, the immune behavior of the tumor. And this description, which is evolutionary conserved, is in bacteria. Paul Modrich did a lot of data, and Richard Colodner did a lot of data on yeast. Won a Nobel Prize in 2015. This is how it looks. So 2 compare with 6, 2 compare with 3, and MLH1 pairs with PMS2. So here's a single mismatch. They act as heterodimers. They actually form a ring around the DNA and can detect the change of shape of the double strand of the DNA. They automatically bind, but much like actin and muscle, they require ATP to be hydrolyzed to come off, and then requires the formation of LH1 PMS2 dimer to signal either repair or apoptosis. The 2,6 repair will find single base pair mistakes, or typically mononucleotide repeats like an AAA or TTT. The 2,3 combination finds dinucleotide or longer microsatellite repeats, so like CA or longer. Here's a tetranucleotide repeat, and I call it the broken zipper model, meaning that somehow a piece of zipper got stuck out and it sticks out like this, such that on this side you'll have 10 repeats and this side you'll have 9 repeats. These proteins will find that, ultimately to exonuclease function, take out from the newly synthesized strand that piece and try to resynthesize through normal processes the correct number of repeats on the other strand. If this repair is taken out, obviously you've now created a frame shift mutation, and this is what we call microsatellite instability. Now the cell, if it signals and the repair processes are there, and it can repair, the cell can go on and live for another mitotic cycle. If it's overwhelming, the cell could signal for cell death. Obviously, if these proteins are missing, you're replicating defective lengths of these microsatellites, as well as single base pair mutations in subsequent cells, which can drive the tumor. So what are the common features of classic DNA mismatch repair? As I mentioned, it's about 15% of all tumors. They display this microsatellite instability, and in a pathology lab, they will lose the DNA expression. For colorectal cancers, it's more common in the right side of the colon. Okay, 70% of all MSI tumors are on the right side of the colon. They often show poor differentiation despite their better outcome for patients. As I mentioned, they're hypermutated. They're diploid. They often lack p53, unlike the chromosome instability tumors. And the tumors attract these T cells. And if I showed you a histology, you'd be amazed because they'll look like almost like a granuloma reaction under some of these tumors. And a good pathologist can spot and say, hey, that's probably an MSI high tumor. That's what Jeremy Jass did when this was first being recognized. But it does make them more susceptible to PD-1 blockade. I'm not going to show you data, but they are more resistant to 5-FU, which is our standard baseline treatment for colon cancer therapy. And they also have long survival, as I mentioned. So the tumor infiltration was first really noticed by Jeremy Jass and some others. And this is a paper from 2008 by a German group named Schweitali, who were looking at these T cells, and they took T cells from these MSI tumors, and they created the predicted frame shift because they had coding microsatellites, and took the T cells and see if they reacted to those artificially made frame shift neoantigens, for essence, or peptides. And the MSI, the T cells taken from a patient with MSI tumor recognize them, but there was no activity from T cells taken from a microsatellite stable tumor. Then they took T cells from the peripheral blood, and the T cells responded to those same frame-shifted peptides if it was from an MSI patient, not from an MSS patient. So this is from the blood now, but also from Lynch syndrome patients because they have a germline mutation in this. But really, the fascinating piece of this was a Lynch syndrome patient who's never had a tumor actually had T cells already made against these frame-shifted peptides, suggesting that they're auto-immunizing themselves. You must be making a small amount, much like you get an immunization against chickenpox or something, and you develop memory T cells such that when you get the tumor, they really infiltrate into the tumor. This, again, was Jeremy Gallon's paper now in 2016, so 10 years after he first did that description, and shows that patients who have MSI tumors develop a lot more memory T cells than microsatellite stable tumors. And if you look at what happens if you have what he calls the immunoscore, and it turns out the high immunoscore, the low immunoscores are less likely to be microsatellite unstable, and the high immunoscore were mostly MSI ones, that all the high immunoscores acquired PD-1 expression on the surface of their cells, such that they're much more susceptible to a blockage. And so this is the pathway that's now kind of generally described, where you go from a normal to adenoma. And if you are one of those 15% or a Lynch syndrome patient, you will lose mismatch repair in there, but you also still have to take out the Wnt signaling pathway. About 25% will get a BRAF. You rarely see BRAF in a chromosome unstable pathway, and you don't see it in a Lynch syndrome. It's only in the sporadic ones. And then because of loss of mismatch repair, you get this hypermutable phenotype. You take out the coding microsatellite genes, and those, because now they're truncated, they form neoantigens that are immunogenic to the immune system. And with the increasing somatic frame shifts and expression of PD-1, these T cells are protecting the tumor, in one sense, from spreading. Now, you can get tolerance and everything, but that's why I think many of these patients do well, even with MSI tumors. Stage per stage compared to someone who doesn't, they will last much longer unless they have some escape mechanism from this. So the pathway is you alter DNA mismatch repair. It could be a genetic or sporadic with MLH1. You get these target gene mutations. By the way, most of these immunogenic peptides are mononucleotide sequences. There's a handful with dinucleotide repeats, almost none with encoding. There's some on promoter sequences with triplicate repeats, and there's like 0 genes with tetranucleotide repeats in their coding sequence. And that becomes important when I talk about EMAS. So most of these are mononucleotide repeats in these coding microsatellites, and then the biological consequences, the protection or reduced metastases in general. There are 5 conditions in which you lose mismatch repair. The most common we hear is this autosomal dominant Lynch syndrome, which you can take out in the germline, any of these genes. EpCAM happens to be on the same chromosome as MSH2, and when you lose it, you secondarily take out MSH2, even though it's genetically normal. For MSH3, which I'm going to talk about next, there is no observable phenotype, and that's because of its biological profile, and I'll touch base on that in a minute. So you get the germline mutation in Lynch, followed by somatic mutation. This is 3% of all patients with colon cancer. And here's the age and the types of cancers they get. And the clinical caveats is that we have to survey and watch these patients very carefully because not only they can pass them to the next generation because it's a germline defect, but we have to watch the patient for the rest of their life to help prevent them from getting cancer. The other inherited form is constitutional mismatch repair deficiency. This is autosomal recessive. This is when a person acquires two bad mismatch repair genes. The most common are MSH6 and PMS2 because they tend to present older and the families don't know that they carry the genes. These are rare. There's about 100 to 150 of these cases reported in the world literature. But you'll get biallelic 2 mismatch repair germline mutations, and they will present cancers as early as 1 or 2 because they have lost mismatch repair from the start instead of one gene and then take out the second gene somatically. And they'll have hematologic and cafe au lait spots as well. About 4 or 5 years ago, the first biallelic MSH3 family was recognized, and there's about 5 of these. We described one of them as well. This is a more subtle phenotype because MSH3 by itself as a monoallelic takeout, there's no observable phenotype. And I get these calls like, I got an MSH3, someone tested it. I'm like, don't do anything with it. But if an MSH3 person marries with another MSH3 person, 1 in 4 of them could get a biallelic loss of MSH3. They tend to present in their 30s. So with one gene, maybe they're dying in the 80s and 90s of something. But if you have 2 MSH3s taken out, instead of 2 years or 10 years, they'll be in their 30s and they'll develop an allelic polyposis syndrome. And the way to identify them, you can identify them germline, but their tumors and their normal tissue would have lost MSH3 from the get-go because they have biallelic loss of that. So that's extremely rare. And like I said, there's only 5 cases reported in the literature to date. The other 3 are sporadic conditions. So the classic is sporadic MSI, that's that 15%. This is biallelic somatic methylation of MLH1. That's 15% of all cancers. They tend to be older, and as I said, in the right colon. And as we age, we tend to get more methylation and possibility, and they tend to occur in the right colon, and they're slightly more common in females, and as I mentioned, 25% are BRAF. There's also a syndrome called Lynch-like syndrome. So they don't have a germline mutation, but they get biallelic somatic inactivation of mismatch repair genes. This is just under 1%, so it's a little more common than you think. And so you'll see this patient, they clearly have MSI, but they won't have a germline mutation. And so, this is about 2/3 of all DNA mismatch repair germline negative patients. They tend to be younger, so there's something driving this. Why did they get this? But right now we use surveillance guidelines similar to Lynch. The 5th syndrome is called EMAS. This is also a somatic, and I'm going to walk through this a little more carefully. EMAS stands for elevated microcellular alterations at selected tetranucleotide repeats. I didn't name it, but it's a MSH3 loss. And let me walk you through that. So that's my next topic. And just to highlight again how mismatch repair works, what I've drawn here is we call MSI high, and it's defined by NIH definition as mononucleotide and dinucleotide instability. Okay, now you can get longer repeats. There was this thing called MSI-L for a while, and everyone was trying to figure out what it was. It actually is MSH3 defect. And then EMAS is anything longer. And if you can see where EMAS overlaps, it perfectly overlaps with loss of MSH3 based on its biochemical profile. That wasn't known 10, 12 years ago. That had to be figured out. And we and another group figured it out by doing knockout models and showing that that's what was happening in these human cells. So like I said, 2 compare with 3 or 6, that's with these arrows, and MLH1 pairs with 2. And obviously, if you take out 2, MLH1 or MSH2, you can get instability at all these areas. You take out MSH6, you only get mostly mononucleotide and a little dinucleotide. And if you take an MSH3, you don't get any mononucleotide instability, but you'll get dinucleotide or longer instability. So this is a paper that one of my former postdocs published 10, 12 years ago or something like that, when we were looking at some of these cancers for MSH3 expression. And you can see MSH3 is mostly stained in the nucleus here, and this is a loss of MSH3. But what we were seeing on these EMAS positive measured by this tetranucleotide instability was kind of partial loss from the nuclei. And it was nuclear heterogeneity, because we were seeing this and we were like, okay, Lynch-like, you get two somatic losses. Maybe you're getting MSH3 somatic losses. But when we sequenced for MSH3, we weren't finding any mutations, which was confounding, but yet we were seeing loss of some of the staining. And it was haphazard like this. It was partial nuclear heterogeneity. So just to, and I'm going to walk you through the why that is, but this is observed in 50%. So it's more common. It's the most common mismatch repair defect in all of colon cancer. Okay. And we don't fully understand it. They display this EMAS, so they don't have mononucleotide instability. They have dinucleotide or longer, and we measure this mostly by tetranucleotide. And there's this heterogeneous or reduced MSH3 expression without any somatic mutations in the tumor. They appear distributed throughout the colon, so it's not concentrated in the right or left colon. And they're not hypermutated. Why? And I'll go back 2 slides here, because most coding microsatellites are mononucleotide repeats that generate that immunogenic type of peptide. So if you take out just MSH3, you don't get much mononucleotide. You don't get any mononucleotide instability where most of those coding microsatellites are. However, they are associated with inflammatory cells. And I'm going to walk through that in a minute. And we thought that, okay, we're not finding mutations, but we see some of this inflammation. And so what is going on? But several of the tumors did have inflammatory cells around it. They seem to be sensitive FIFU. We published that paper as well. And they show shorter survival. So I got a different mismatch repair defect in which one, the classic, you get longer survival. But if you take out MSH3, you seem to get shorter survival and poor prognosis. And we published a couple of papers showing that it's higher frequency amongst African-American tumors, which tend to be more inflammatory as well. This is a study from another group showing the difference between EMAS and classic MSI. MSI, very protective on lifespan. EMAS, very bad on lifespan, okay? And this was as statistically significant as differences in stage. Okay, so this form of MSI is not good. This form of MSI is much better. This is a study we published, again, 10, 12 years ago, looking at a racially diverse group of rectal cancers. And we showed that there were twice as many African Americans who had EMAS positive rectal cancer. They were associated with advanced stage, as I mentioned, and most of them had, or at least 2/3 of them had, this peritumoral inflammation around the glands. So they had the intratumoral cells and this peritumoral inflammation that seemed to be intimately associated with the glands. So we were thinking like, okay, we can't find a mutation. What the heck is going on? So then we said, well, maybe the inflammation has something to do with the loss of MSH3. So we started looking at cytokines. And here's the punchline here is that when you look at IL-6, which is a pro-inflammatory cytokine, here's the EMAS positive. You can see more IL-6 was associated with more EMAS positive and less was associated with EMAS negative. So we said, we wondered. We also, so then we said, okay, we're going to test IL-6 and we're going to take different cells, give them increasing dose of IL-6 and map out what's happening to each of the mismatch repair proteins. And so you can see we went from control to 1,000 micrograms and nothing happened. And we're like, so we were totally perplexed. I can't find a mutation. I can't find the reduction of the protein, but yet my immunohistochemistry is saying something's going on. So then one of the people in my lab did the key experiment, and that was this experiment. What they did is they went to immunofluorescent microscopy, and then they separated out the nuclear and cytosolic fractions of the cells. It's like taking the egg yolk from the egg white, right? So if you look at the immunofluorescence, so MSH3 is stained in green, DAPI stains the nuclei, and you can see all the mismatch repair proteins in the nucleus where the DNA is. Then what happens, you give IL-6, and the green disperses, and so it is outside the nucleus. And that explained the heterogeneity that we saw. It was associated with oxidative stress bursts, and when she did the nuclear cytosolic separation, separating the egg yolk from the egg white, by giving increasing doses of IL-6, you can see it drop out of the nucleus and transfer to the cytoplasm. And this was unique to MSH3. It didn't happen for MSH6, MSH2, or MLH1. And you can see how the histone fractions with the nucleus and the alpha tubulins with the cytosol. In addition to this, we took a native tetranucleotide sequence in the cell and just gave it IL-6 and see if it frame shift mutated. And after 2 weeks, there was 10-fold more frame shift mutations after IL-6, suggesting that if it moves out of the nucleus, it's not repairing as the cell replicates, and you're getting more frame shifts at those tetranucleotide sequences. We also looked at other pro-inflammatory cytokines. None of them moved MSH3. So it was unique to IL-6. So just for most people, you probably know the IL-6 pathway hand backwards and forward, but IL-6, you can have classical and trans signaling, and it goes down a GP130, does the JAK-STAT kinase pathway, and then signals transcription in the nucleus. And so we use various things, anti-IL-6 antibody, soluble GP130 to prevent it from binding, and a drug to prevent STAT3 from dimerizing to see if that truly blocked it. And that's exactly what's shown here. Here's IL-6, it disperses. You give anti-IL-6, it doesn't disperse. Soluble GP130, it doesn't disperse. And the drug to block STAT dimerization, it doesn't disperse. And you can see that here also in the nucleus cytosolic fractions here. IL-6 moves to the cytosol, but if you block it with anti-IL-6, soluble GP130, or the drug, and it doesn't move. It blocks most of the IL-6 movement. Then we put a constitutionally active STAT3 in it. So I said, okay, if it goes down the JAK-STAT kinase pathway, if you just jazz up the STAT3, what happens? And so we did a transfection experiment, and you can see which ones got the MSH3 flag, okay? And you can see where the MSH3, and you can see that in the cells that actually got the MSH3 that didn't get the MSH3 flag, you can see a very sharp nucleus. But in the ones that have the MSH3, the nucleus is much more fuzzy. And here you can see that here you put this the constituent active STAT3 here. It moves the MSH3 to the cytosol by itself, even without IL-6. So then we're saying, like I said, why does this move? And MSH6 doesn't move, MLH1 doesn't move. Why does MSH3 move? And so no one had characterized the protein. So Stephanie Zing-Roginski, this was her big project. She analyzed the MSH3 protein. And by looking at conservative regions, theorized that there were 2 nuclear localization signals possible in there and up to 3 nuclear export signals on the MSH3 protein. She mapped those out and then deleted each one to see if, what would happen. And so here's 2, this is, we call it at that time, NLS1, NLS2, and it turns out when you, here's the wild type, you take out the NLS1, it automatically is stuck in the cytosol, okay? The NLS2 did not. So we knew that NLS1, what we called initially, was the bona fide nuclear localization signal. We also did the same thing for the nuclear export signals. There were 3, one of them didn't pan out. So we were down to 2, and it turns out those 2 cooperate with each other to move it back into the nuclear export. I'm sorry, nuclear export. So if you take out one or the other, you can't move it, and if you take out both. So it was only if you had both in that after IL-6 that it actually moved to the cytosol. So we mapped all that out. Now, I got even more complex than that because this is the MSH3 protein. These are the main activity domains. Remember, I said it has an ATP domain. And it turns out the nuclear localization signals in the N-terminus of the protein. And at that very beginning of the protein, there's a polyalanine proline-rich signal just proximal to the nuclear localization signal where we mapped. And you can see the 2 nuclear export signals that we mapped. We just looked at, like, okay, since this is known to, this polyalanine proline-rich region are known to be polymorphic, we just looked at a bunch of colon cancer cell lines we had in the lab, and you can see that it is polymorphic. But we noticed there was this one delta 27 that kept popping up in a bunch of the cell lines. And then we characterize all these cell lines. The delta 27 actually has 27 base pairs by shortening this alanine and proline-rich region. And we noticed that, okay, so if you take this cell line, which has full length, for both of it, here's control, here's IL-6, so it moves it to the cytosol. And if you give hydrogen peroxide, remember this was associated with the oxidative stress burst. The oxidative stress doesn't do it, it's really the IL-6 signaling. But if you look at one that had delta 27, the IL-6 moves it, but oxidative stress also moved MSH3. And again, it didn't move any of the other mismatch repair proteins. So we were a little perplexed by that. So we engineered cells with MSH3 that had delta 27 and non-delta 27. And you can see here, and this is matched by the immunofluorescence, that here's untreated, here's full length, delta 27, and untreated, it's all in this nucleus. Then if you give IL-6, both the full length and delta 27 move. But here you give the oxidative stress, only the delta 27 moved, but not the full length. So again, even with the engineered cells. And when we did a timing experiment on this, I shouldn't say timing, a dose dependence experiment, the delta 27 just became way more efficient in moving to the cytosol, or at least getting stuck in the cytosol, I should say, than the wild type. So there was something funny going on with the delta 27 version. And then when we did the computer modeling of this, so this is the full length MSH3. You can see the map, the nuclear export signals, but the nuclear localization signals is on this long piece, and it's right at the tip of this. And you can imagine some protein grabbing that and moving it. But when you have the delta 27 changing the length of this polyalanine track, the nuclear localization signal moves from the tip to the side where it can't efficiently get back into the nucleus. So we think that that delta 27 is prone, and this is in the population. So when we map this out amongst patients with colon cancer, this is 171 colon cancer patients genotyped for Delta 27. And compared to what's in the UC Santa Cruz database, it came out to 0.0012% of the population based on the UC Santa Cruz. But yet we found it in 1.2% of these colon cancer patients, which was, 1,000-fold higher. So we're like, well, maybe if you somehow get this genotype, maybe you're more prone to this more aggressive phenotype based on, our mapping of this. We also took this to the next level. I said IL-6 moves this. What disease is better known to IL-6 than inflammatory bowel disease, okay, or any inflammatory disease? So we talked to our IBD folks and we got some UC tissues. This is ulcerative colitis. And we were surprised to find that some of them, when you stained for MSH3, actually lost nuclear expression. They did not lose MSH6 expression. So we said, maybe there's something to this. Okay, so then we mapped it out by the markers. So mononucleotide, dinucleotide, and tetranucleotide markers. So here's normal tissue. There was 10. So there should be no instability at mononucleotide, dinucleotide, or tetranucleotide. So that was good, okay? Then we looked at 70 UC tissues. So these are non-cancer, and they're theoretically quiescent in inflammation, but they're UC patients. Now 70 cases. There was no shift of mononucleotide, again, suggesting this is MSH3, but yet we found shift of some of the dinucleotide markers and the tetranucleotide markers. Then we looked at it and said, okay, let's look at a handful that we'll find some with dysplasia and cancer. So we did that. So so keep in mind, you're seeing genetic shifts in normal tissue without any cancer or precancer, okay? So now we got dysplasia. We have found a handful of those. And again, no mononucleotide all. They were only dinucleotide, or in this case, the tetranucleotide. We even looked at different stages and then early stage sporadic cancer. The interesting thing is, as you move from inflammation to dysplasia to cancer, the percent of shifting went up, suggesting that this was an acquired longevity thing. When we looked at a non-UC sporadic cancer, we found mononucleotide instability. We just picked 18, suggesting that this is clearly an MSH3 driven process in UC. Then we looked at, okay, let's look at short-term UC and long-term UC. And in short-term UC, the amount of EMAS was 16%, but in long-term, this is people who've had it 8 years or plus, that's when you start worrying about cancer, it jumps up to 40%, suggesting that this is accumulative over time. So we have genetic variations that are happening in the setting of inflammation, even before cancer, that might maybe predispose to cancer. We also looked at these samples, again, going back to the UC Santa Cruz database, very few people have this Delta 27. We found 15,000-fold higher of this Delta 27. So it might be that some people are just a little more predisposed if they're carrying this polymorphism available that can lead to more inflammation and worse disease. So just to summarize this piece and see where I am, I got 7 more minutes. I might stop here and not go to the third piece. But basically, however the inflammation happens, you get IL-6 signaling, and now you have this MSH3, not the other mismatch repair proteins, that via a nuclear export signal can shift out into the cytoplasm. If it's Delta 27, it's harder to get back in because you've altered the ability for the nuclear localization signal to come back into the nucleus, okay? And of course, if you're in the nucleus, you'll have repair, but if you're not in the nucleus, you don't get repair. You got frame shift mutations occurring, and the EMAS formation occurring. And so the way these tumors are forming is that you start perhaps with some initiating event, you get inflammation, however that happens, but that's IL-6 driven to lose the function of MSH3, not by mutation, but by shifting it out of the nucleus into the cytosol. And we see this in 50% of colon cancers. So there's some functionality to this. Now, what happens downstream of this is that you can get these frame shift mutations, but it turns out MSH3 along with MSH2 is part of the homologous recombination pathway. So they can fix, and these are studies that were published in Cancer to Study. There's about 6 of these studies implicating MSH3 in double-strand break repair. And so that may be a driver of this, because most of these frame shift mutations are not encoding microsatellites and don't have the same immune function like when you take out classic mismatch repair. But the biological consequence of that is that you're worse off by having this inflammation-induced EMAS generate. So should I stop there or should I do the last 6 slides? I'll leave.
It would be maybe a couple minutes because it would be interesting to have a
Okay, so I'll go really quick on this. I'll just say this, that, you know, if this is like my first slide, it can happen anywhere in the tumor progression or initiation. So that can modify this. We know that the microbiome is intimately associated in the colon. I mean, we have the highest level of microbiome in the colon. And even with neoplastic lesions, there's been association of certain types of microbiome. The most prominent have been in early neoplasia, Colobactin producing E. coli and an enterotoxin B. frag. They both make a toxin. There's strong evidence associated by epidemiology and microbial enrichment. And in late neoplasia, Fusobacterium, and it also does make a toxin. And just this is Cynthia Sears' work. She took FAP patients, which make a lot of polyps, and controls that don't make polyps. And she shows that if you have the Colobactin producing E. coli or enterotoxin B. frag, and certainly both, they're more associated with people that have polyps than with not polyps. And then if you take the mouse version of the FAP, the APC mice, and you give them these organisms, here's the E. coli, here's the enterotoxin B. frag. But if you give both of them together, you shorten the lifespan of the mouse. And you can see that the inflammation score goes up, particularly with both of them. And if you take an IL-17 knockout mice, and IL-17 helps drive IL-6, that you don't get the tumor formation, suggesting that this inflammation can be driving that. She also showed, and this is from human colons, that Fusobactin shows up in the tumors almost exclusively in the right colon, where we think of MSI high tumors, or right-sided. And the risk of proximal colon cancer with this Fusobacterium is 5-fold higher if they had a Fusobacterium containing biofilm there. This was associated with IL-6 generation, IL-6. If you have biofilm, you see it's elevated versus no biofilm. And then also STAT3, as I mentioned, much more elevated. And you can see that by staining with PCNA, the proliferation of the normal crypts is higher in the presence of the Fusobacterium containing biofilm, okay, compared to those that did not have the biofilm, suggesting there's some synergism between these organisms and the growth and maintenance of these tumors. I'm gonna skip that one. Fusobacterium, so there's hardly any studies on how this affects DNA, because we're like, well, you can lead to inflammation, but how does it affect DNA? And this study, which we just published a couple of years ago, we just took cells, threw some Fusobacterium, took the supernatant in it and see what happens. And it turns out the supernatant can cause double strand breaks. which was amazing. Presuming from the toxin or something from the bacteria. So they picked up gamma H2X stains, new ends of DNA. And then you can block it by giving metronidazole in the thing because that kills the fusobacterium. I'm just going to briefly say that we looked at associations with the genetic pathway. It turns out if you have fusobacterium, this was 304 cases we looked at, that it's strongly associated with the MSI high pathway. It is also associated with the EMAS pathway, but less so than MSI high. Comparable, it's more than twice for MSI, and it's strong associated with MLH1 hypermethylation. And the bottom line is, I'll skip these slides, that it was mostly strong associated with that CpG island methylated phenotype pathway, which is the methylation pathway that gives you serrated adenocarcinomas on the right side. So that was, so the last thing I'll say can affect chemotherapy. So this is not my work. This is Matt Myerson's work when Susan Bullman was there. She's now at the University of Washington, and she published this incredible paper in Science looking at fusobacterium. These are two examples of patients from a series they had in which they had the primary tumor and the paired metastases in the liver. So this is the primary tumor here, and they did RNA in situ for fusobacterium. So you can see the fusobacterium is in this pink. It's stuck, intimately with this tumor gland right here. Okay, here's another one. You can see it very intimately with the tumor gland. That's the fusobacterium. It's a minimally invasive bacteria, right? Then in the Mets, they also confined in the Mets, in the liver, fusobacterium stuck to some of the glands. So the $64,000 question was, did the fusobacterium follow the metastases to the liver, or did it recolonize after the metastases set? And so what they tried to answer this with the serial passage experiment over 3/4 of a year. They took mice, then took it out another mice, passed it another mice, passed it another mice 8 times. So you passed it 8 times, and they could culture it each time through at least 4 passages through these mice, and the PCR could be detected all the way through. So their suggestion is that it went with the metastases to the liver. So the bacteria is following the metastasis. That's their hypothesis. So they took these are cells put into mice, and these are HT29, which has no fusobacterium. Metronidazole does nothing. But when they take the PDX, a human PDX that had fusobacterium with it, and they give metronidazole, they shrunk the tumor by 30%. Okay, suggesting that the fusobacterium is producing something that is helping that tumor to grow and survive. So just to close, I just highlight defective DNA mismatch repair is an important process for repairing DNA. You can classically take it out by somatic inactivation, most commonly by methylation of MLH1, or you can have Lynch with germline mutations. You get these new point mutations, frame shifts. There's 100,000 of these non-coding, but about 500 encoding genes that give you immunogenic, and you can get immune checkpoint susceptibility because of that process. But there's another process called this inflammation that triggers this MSH3 shift that we still don't fully understand. This causes frame shifts at these longer microcellulites, but not the mono microcellulites, and possibly cause double strand breaks and changes the outcome of the patient. So I think of it this way. I showed you this slide before, but EMAS seems to be a modulator, not a direct pathway, because if you take out MSH3 by itself, it doesn't drive tumors, but it seems to modulate the outcome. And in my model, I put it as a modulator for all 3 pathways. So I'll stop there and hope that was enlightening. I hope that last part was okay too. Sure.
Great talk. I was wondering, for the IL-6 story, you had mentioned T cells. Were there other immune
The
cells?
which story? I'm sorry.
The IL-6.
Oh, sorry.
IL-6 story. You mentioned T cells. I was wondering if there are other immune infiltrates cell types there besides the T cells.
So I believe there are. So we didn't characterize all the cells. I think Jeremy Galone has been more the expert in that, and he's been genotyping a lot of these different types of T cells. But I can tell you there's a predominance of CD8 positive T cells that seem to be, they seem to be much more correlated. We've published a paper that it seems to be more the CD8 versus any other type T cell, but there are other immune cells there as well. So you're absolutely right. So it's not, I can't tell you the whole portfolio of the immune cells there, but that one seems to be the predominant one interacting for here for some of those neoantigens.
When they do colonoscopy, they concentrate on the polyps.
Yes.
How are polyps correlated to your study? And have they been studied in correlation with what you were doing?
So I assume the question is there, I presume, EMAS in adenomatous polyps before the cancer? Is that kind of what you're asking? Or?
Yeah. My question is usually when they do colonoscopy, when they see polyps, they remove.
That's correct.
Yeah. So they are pre-cancer. Am I right?
Yes.
Yeah. How is it correlated to your study?
Okay. So we did look at about 30-something adenomatous polyps. So it's variable, the expression of EMAS in them, because not all of the polyps are fully inflammatory. So EMAS can be found in polyps. And typically they're found in polyps that have much more inflammation and sometimes ulcerated. So we published a paper on that. That seems to be more correlation. So I can't tell you that EMAS is the driver of the polyp to the cancer, but in some polyps, it might be. It might be. It's not in all polyps. If that's what I don't know if that answers your question.
Okay. When they do
So, yes.
Do they take?
We take out all the polyps.
Samples,
So
to see if they are predisposed to future cancer.
Not normally. So normally at colonoscopy, for screening colonoscopy, we do take out identifiable polyps. We don't usually take random biopsy unless there's a reason to do that. There are some patients you would take biopsies, but that's not for screening. And so we have analyzed polyps. We have not analyzed, other than the samples with like ulcerative colitis, we have not analyzed a bunch of just normal samples. I'm not sure if that's what you're asking.
Okay, what I mean is that fusobacterium, can they?
Fusobacterium. Yes.
Locate fusobacterium in different regions
So,
of the colon to see if they.
Yeah, you can. So that was Cynthia Sears' work. That wasn't my work, that particular study. So she did map, she took random samples. So that was a design study where she'd had FAP patients and non-FAP patients and was able to take biopsies even of normal tissue and could show that if the presence of fusobacterium was there, the risk of developing a cancer in the long term was 5-fold higher. And so it could be on the normal tissue. And some of the immunohistochemistry I showed you was some of the normal crypts were more proliferative with the presence of fusobacterium. Now, fusobacterium is normally an oral bacteria. We still haven't figured out how it gets from there to there. So that's another question. So do we swallow it? You brush your teeth? Does it spread hematologenously? But it's an anaerobic bacteria. So the best place to live in the colon is actually the right side of the colon, where it seems to do work, because that's the most anaerobic part of the colon. So it has been found on normal tissue, yes, and it can be associated with increased proliferation, but its most strongest association is typically with a cancer. But she did find it on normal tissue with biofilms. Does that answer your question?
Somewhat.
Okay.
Thank you so much. We're out of time. And I know Dr. Carethers has other people to meet and people to see. So thank you so much for attending. And thank you so much for a wonderful lecture. It was really fascinating. I mean, I'm sure there's many, many questions I had in my mind, but we don't have time. But thank you so much.
Well, thank you. Thank you
everyone.
for having me.